very good afternoon to you. I'm Howard Feldman, and this is the Synthesis Podcast. Each Sunday, as you know, for the last 5 or maybe even nearly 6 months, I sit down with Dr. Anton Meyberg, who's a COVID expert, and we talk about the latest development in the pandemic and how we are doing around the world as well as here in South Africa. We answer your questions. You send them through to us, and I pose as many questions as possible to Dr. Meyberg. The one recurring question and the one pressing issue is where we are with regard to a vaccine. How are we doing globally, and when is this battle likely to be won? You spoke and we listened. Joining us today is Brian Abel. He's a PhD, MBA, MSc. He's a immunologist. He's a lot of different things, and he is very involved and at the forefront of vaccine research, and specifically the COVID-19 vaccine research. I've invited Dr. Anton Meyberg on this to this podcast as well, because one of us at least needs to know what it is that we're talking about. Good afternoon. This is the Synthesis Podcast. I'm Howard Feldman. Brian, good afternoon to you. Anton, good afternoon.
So good afternoon, Brian. Good afternoon, Howard. And good afternoon, everybody. I think today's discussion is extremely relevant and will hopefully be very thought-provoking. Our aim is to have a discussion that explains the hows, the ifs, the whens, but also to extrapolate relevant data. And this will hopefully clarify where we stand in the race for the vaccine. In order for a vaccine to undergo development, we first have to prove the vaccine works and that it is safe and effective. Then the vaccine needs to be made and ultimately used. In essence, this development process normally takes about 5 to 10 years, if not longer, and costs about $1 billion US, with a failure rate of about 93%. We are now asking the world to come up with a vaccine in 12 to 18 months. Never been done before, but we're at a stage that if not now, when? Another burning question is, will we be able to use it with equity and give access to all? Can it cause a significant reduction in the COVID-19 burden and disease to alleviate the suffering that this pandemic has brought? And why are we so confident that we will get the vaccine when we haven't been able to find a vaccine for HIV? The key to it, as the Americans say, is that they are now working on Operation Warp Speed, making a vaccine that's already in phase 3 trials, which will financially be underwritten by the United States government. Do we believe the Russians, or have they cut corners by not fulfilling the criteria for phase 3 trials by testing it on less than 30,000 people? We have burning questions. We have an urgent need to get a vaccine so that life can once more return to some sort of normality. But will it ever? Brian, maybe you can start by explaining to us what a vaccine is, how it works, and you can explain the brilliance of the fact that the virus genome was sequenced and 8 weeks later, vaccine testing was already underway. What are the South African vaccine trials, and what does the near future hold for us?
Hi, Anton. Thank you very much for that framing, and you put in a really, really interesting perspective. We're certainly at a really crazy phase at the moment with the COVID-19 pandemic, and a vaccine is urgently needed. A vaccine is an agent which plays a role in inducing protection in a host. It can take lots of different formats. One of the classical formats for a vaccine is actually taking the organism itself and weakening it, so having a so-called attenuated vaccine, and then giving that to the host, which then induces the protection without actually causing disease in the host, but the host actually gets an immune response which then protects against exposure to the organism itself. You can have lots of different formats of vaccines, and I'm going to touch on this during my presentation. You can have protein vaccines, which incorporates the relevant, what we believe are protective parts from a virus, for instance, which are then injected into a host and induce a response. And you can also have vaccines which are virally vectored, and they can be, for instance, a yellow fever vaccine, which is known to be highly efficacious, induces about 99% protection. And you could take a yellow fever and you could incorporate, for instance, the SARS-CoV-2 virus components into it, and this can then be injected into a host and induce protection. And I'll touch on a number of these during my presentation.
All right. So, Brian, if you would maybe take us through the presentation, and obviously Anton and I, where I possibly can will ask you some questions and in order to seek clarity.
Okay. Thank you, Howard. So I'm going to take you through a number of slides on the topic. So I think a number of you will be quite familiar with this particular format. This is actually the Johns Hopkins University Coronavirus Resource Center. The data was pulled from last night, and it shows that we really have a serious pandemic on hand. We have almost 23.5 million global cases. I think today we're over 810,000 global deaths. And in the context of South Africa, you can see we have a hotspot shown over here in yellow. And I think the number of deaths has now actually exceeded 13,100 as of today. So we have a serious pandemic on hand, and there's really an urgent need for a vaccine. So a resource that I use and I'd highly recommend to all of the audience is the COVID-19 tracker from the Milken Institute. You can see the URL in the bottom right-hand corner if you want to go there yourself. As you can see, there are 316 treatments under development at the moment and 203 vaccines in total. This is really an unprecedented scenario where we really have an enormous amount of treatments which are rushing ahead expeditiously with funding, as Anton mentioned before. And if we focus on the vaccines, actually 26 of these are already in clinical trials, of which 6 of these are in phase 3. So it's really incredibly fast how these vaccines are moving through the clinical trial stage. What does one normally need to do to get a vaccine? And shown over here is an ideal vaccine setting. If you focus on the top, you can see it starts with research and development, where one chooses a platform choice. And as mentioned, it could be an attenuated or weakened virus. It could be a protein subunit. It goes through different stages where you select targets. In the case of SARS-CoV-2 or COVID-19 infection, most vaccines are actually taking into account the spike protein, which I'll touch on later. And this is the protein that is actually dotted on the surface of the virus envelope, and it's used to actually enter the host cells. After validating the target, there's movement to a preclinical phase, which is mouse models, and it can also incorporate monkeys. And once this is shown to actually be efficacious in an animal model and also not toxic, it can move through into a phase 1, which incorporates safety in healthy individuals, phase 2, which looks at safety but also efficacy, and phase 3, which I've mentioned 6 candidates have already moved into. And at phase 3, one is looking at tens of thousands of individuals, so you can actually ask the question around protection. In the ideal setting, if the vaccine is shown to be efficacious and also not toxic, it moves forward into the marketplace. And in the ideal situation, one would hope that you could have billions of doses, you could elicit effective, long-lasting immunity. And I think the reality of the situation is that not one candidate is actually going to suffice to do this. We're going to need a number of candidates to be able to vaccinate a world population of 7.8 billion. As Anton touched on before, a normal typical vaccine development takes somewhere around 10 years. It could exceed this. The expectation during the pandemic is 12 to 18 months. And, you know, we're going to have to see how this actually unfolds. And to put this against historical background, if you look at a number of vaccines that have been developed and their timelines, you can see for polio, one needs to take into account that this is more than a century ago. But the vaccine developed by Sabin took about 60 years to develop. Ebola, which was also fresh in our minds, and you know, there've been a number of epidemics in West Africa recently. From the first vaccine being published in 2005, it took about 15 years. For other coronaviruses like SARS and MERS, it took about 6 and 17 years. Went into clinical trials, but the actual epidemics were extinguished, so no vaccine actually went into the marketplace. For SARS-CoV-2 or COVID-19, we're hoping that we'll have a vaccine in 12 to 18 months. Another important aspect to take into account is with the expeditiousness of making a vaccine, there's a concern around toxicity, or that vaccine might induce, lead to enhancement of disease. And this is the case for dengue infection as an example, and also has been shown in animal models for other coronaviruses. So shown on the left-hand side here is the COVID-19 vaccine design and the Janus face. On one hand, you can have protection. On the other hand, you can potentially have toxicity or immunopathology. If you look on the right-hand side, you can see different vaccine formats. And if you're able to induce an appropriate immune response, this may look like a B-cell response inducing neutralizing antibodies shown over here, where the neutralizing antibodies can bind to, for instance, spike protein on the virus and prevent infection events. Also, we would hope that an appropriate response would include T cells, like killer T cells, which would eliminate infected cells. On the right-hand side, you could have enhancement of viral infection, where an inappropriate response leads to binding antibodies, which can actually enhance uptake of the virus. Examples of this were, for instance, the STEP trial in HIV, and also dengue. It's been shown that you can actually enhance infection as well. So what does a natural response look like, and what should we learn in order to make a rationally designed vaccine? So shown in the top here is the SARS-CoV-2 virus, which actually causes COVID-19 infection. And I've once again over here highlighted the spike protein, which is the one that is actually used for entry of the virus into the host cell. I'm not going to take you through this very complex diagram over here, but what you can see is that spike binds to the ACE2 receptor on the host cells, for instance, in the lung, and this leads to infection. Most vaccines are actually inducing B-cell responses, which lead to ideally neutralizing antibody responses, which can then prevent infection. But at the same time, there's the potential to also induce a cellular response, such as a T-cell response, and shown over here are helper T-cells, which actually play a role in also driving a more appropriate B-cell response, but also they're very important for persistence of immunity. And the cytotoxic T-cells or killer cells are involved in eliminating infected T-cells. So I think it stands to reason that to have a really effective vaccine, we'll actually need to have both arms of the immune response, not just antibody response, but also T-cell response at the same time. At the moment, there are really a large number of different vaccine formats that are moving forward. As mentioned, there are 203 under development. In the pie chart, you can see that there are 9 different vaccine formats shown over here. And if you look at the syringe in the middle, this is from the Milken Institute, you can see the 9 broken down into the different colors. And as you can see, for a protein subunit vaccine in orange, and also for the DNA-based vaccine, a number of candidates are moving into phase 2. At the same time, inactivated viruses, also RNA-based vaccines and non-replicating vaccine candidates are also moving into phase 3. So, you know, there's an enormous amount of vaccines moving forward in different formats. But a lot of people are asking a question, what is herd immunity, and also what effect will a vaccine have on transmission in COVID-19? So as you can see, if you focus on the video on the left-hand side, you can see in a naive, so unimmunized population, and in the top left, you're looking at 0% vaccinated all the way through to 95% vaccinated in the bottom right. You can animate this and you can see what a naive population would look like as opposed to a population that's been 95% vaccinated. And you can see that you have very few transmission events here if you have a very efficacious vaccine with 95% vaccinated. On the right-hand side, you can model this in the case of COVID-19. And as you can see in the top over here, if you assume a reproduction number of 2.5, which means that one person will infect 2.5 people with this particular infectious level, you can see that you require 70% effective vaccine with 3/4 coverage to lead to 100% case reduction if 0% of the population is infected at this point. Whereas if 5% of the population is infected, when a vaccine is ready with the same reproduction number of 2.5, you need 80% effectiveness and half of the population to be covered to reduce the peak cases by 80%. So you can see there are a number of factors one needs to take into account, not only the infectiousness of the vaccine, but also the effectiveness of the vaccine and also how many people are actually vaccinated in order to have an impact on the COVID-19 pandemic. Also importantly for a vaccine format is not only how good it is and how effective it is, but also how easy is it to manufacture and scale up. As you can see over here, the Milken Institute has put forward 3 scenarios. On the left-hand side, you have a non-ideal case, which is a vaccine going through all the way through to phase 3, but it doesn't meet regulatory requirements, such as not being efficacious or being a little bit toxic. In this case, it doesn't move forward and there's a waste of time and resources. In the middle, you have a good case where a vaccine actually goes through regulation, but unfortunately, the vaccine, there's no potential to scale up the vaccine, so it's going to take a long time to meet the requirements of a global population. On the right-hand side, you have a best-case scenario where a vaccine actually goes all the way through to phase 4, so it's out in the market, and there's scalable potential. So this would then more ideally meet the needs of a global population.
So Brian, so far I'm following you and everything's making complete sense to me. And the whole point of what you said, that the polio vaccine took over 60 years to develop. The World Health Organization today has declared Africa free from polio. Now, this is more than a century later. We are now working on getting this vaccine out for the coronavirus, for COVID-19, and we're hoping within 12 to 18 months. And there's no data to say that if we give the vaccine to many people, how long will it take to get this immunity? What are your thoughts on this?
It's a very important question. And Anton, I touched on earlier the modeling with taking into account a certain infectiousness of the organism. And this is something, the reproduction number is also changing. Because data is being gathered, and I think it also differs according to the population, according to the susceptibility of the population and the socioeconomics, and how close a population is living together. So, one will need to kind of model this for each nation, as opposed to on a global stage. But, really, I think with the modeling, one will really need to have about a 75% efficacious vaccine if you only have 3/4 of the population vaccinated. And I think we can assume that depending on the vaccine format, one would hope that in about a month, one will have protection if the vaccine format is able to induce with a single shot. Some other vaccines might require boosting, such as the RNA vaccines and maybe the protein subunits, and this might then require another month. So I think you're looking at 2 to 3 months from the time of the vaccination. And of course, not everyone is going to be vaccinated at the same time. So there will be a certain staggering. And then one will need to take into account, as mentioned, how effective the vaccine is and the global coverage of the population. But I think the models show that perhaps with about a 75% effective vaccine, if 3/4 of the population is covered, we can actually start extinguishing this pandemic, and there will be less requirement or no requirement for social distancing in the ideal world. There are a lot of factors to take into account to achieve that, though.
Perfect, thanks.
So just to get back to the presentation, and a lot of people are really interested at looking at where we are with the vaccines and what are the front-running vaccines. And Anton touched on Operation Warp Speed, where the American government has actually committed approximately $10 billion towards the COVID-19 space. I think about 3/4 of that has actually gone towards vaccines. And if you look on the left-hand side, you can see that shown over here are 10 vaccines, 6 of them, as mentioned, are already in phase 3 or moving into phase 3 in the case of BioNTech, Pfizer. A number of these are actually Chinese. Many of those are actually either attenuated vaccines or non-replicating viruses. And then if we look on the right-hand side, these are the vaccines that have been given enormous funding from Operation Warp Speed. And some of the candidates, I think, which are in the public's mind now from the media are the Oxford vaccine, which is covered by AstraZeneca, Moderna, BioNTech, and so on and so forth. And the 2 that I wanted to pick out here are actually in testing or will be in testing in South Africa. And these are the Oxford AstraZeneca vaccine. And you can see they received $1.2 billion to expedite through phase 3 and also to secure a number of doses for the American population. And Novavax is a recent one that was announced that's also moving to be tested in South Africa. This is a protein subunit vaccine, and they got $1.6 billion from Congress. So just to give a little bit of information on these candidates, the first one I mentioned was the Oxford University candidate. So this is known as ChAdOx1 nCoV-19. ChAdOx stands for chimpanzee adenovirus Oxford. And just to give a little bit of information, this is actually derived from an adenovirus, which is derived from a chimpanzee. The reason that we look for vaccines from other hosts is there's a possibility if the human population has got seropositivity, in other words, if they've seen this cold virus before, then there's a chance that they'll actually recognize it and the immune system will eliminate the vaccine very quickly, meaning there won't be persistence and there's a chance that you're not going to get a really good vaccine response. So this common cold virus is derived from chimpanzee, and it's genetically engineered to express the spike gene. So what happens is the spike protein is actually produced in the host. Your immune response generates a response against the spike protein, which then hopefully leads to neutralization of the virus and clearance from the system. The ChAdOx is weakened and it's actually not able to replicate, so it's considered a very safe vector. And the history for this particular format, it's the same strategy as vaccine candidates for flu, TB, and Zika viruses. None of these are yet in the marketplace, but they've gone into phase 3s and there's really good safety data. In terms of the developmental stage, it's moved into phase 2 and 3, and this is underway in the UK, Brazil, and South Africa, and is actually starting at the moment in the US. In terms of South Africa, 2,000 patients will be enrolled in Johannesburg, and they're going to evaluate the efficacy, safety, and immunogenicity in 1,950 HIV uninfected individuals, and then also 50 HIV individuals who have their disease under control with treatment.
Can I just ask you, you've been very involved in HIV research as well. Do you think that this is an important factor in treatment and in the vaccine?
I think it's certainly important. You know, initially vaccines are tested in healthy adult populations, and as one goes through, you need to actually test more vulnerable populations. So once you've actually hit the appropriate dosage for a vaccine, you start going down in age, and then also ultimately you start looking at more vulnerable populations. It could be a population that already has a disease, and it could be an HIV positive population, because at the end of the day, a vaccine is actually ideally is going to be utilized for all people. So you want to really ensure that it's safe to be utilized in the entire population. At the same time, you also want to see that it's actually efficacious in different population groups, whether it's people who have HIV and are chronically controlled, or in other ethnogenetic populations. You may find that a vaccine, for instance, is highly effective. This is the case for TB with a BCG vaccine. It can show anything from 0 to 80% efficacy depending where the vaccine is given. So it's really important that vaccines are tested in different populations, and actually phase 4s which I haven't gone into here, are post-marketing surveillance studies where vaccines are tested all around the world in different populations, and we gather data about how efficacious is a vaccine, and also, you know, is there any concerns about adverse events. So it's very important that a vaccine is tested in different populations and different settings. So just to touch on one last candidate that's been tested in South Africa, this is the Novavax candidate, otherwise known as NVX-CoV2373. So this, as I mentioned before, is a protein subunit vaccine. They use a nanoparticle formulation, and they use an adjuvant called matrix M. And an adjuvant is just an agent which actually allows, it gives the vaccine more potency usually. And this can be through amplifying the immune response and potentially also inducing a depot where the vaccine can persist for longer and induce a stronger response. And this really differs depending on the design of the adjuvant. In the case of this candidate, the protein is once again spike protein from SARS-CoV-2 virus. Historically, there's a lot of safety data around protein vaccines. And upon injecting the spike protein into the host, there's an expectation that you'll induce an immune response, which will probably be robust antibodies and also CD4 T helper response. Important to note is that there are a number of licensed vaccines that uses this approach, and these include, for instance, hepatitis B virus and also shingles. And Novavax themselves have vaccine candidates which are not yet out in the market, but for flu, TB, and also Zika viruses. In terms of where the vaccine is at, a phase 2B clinical trial is starting in collaboration with Professor Shabir Mahdi at Wits University. And this will include 2,665 healthy adults, testing efficacy, safety, and immunogenicity. And once again, as mentioned before, an HIV positive population who are medically stable will be tested with this vaccine, and this will number 240 individuals.
So Brian, let me ask you a question. Yes. Okay. You know, everyone's talking about this Russian vaccine, the Sputnik 5. Is this considered a non-ideal, an ideal, or a best case vaccine? And, you know, are they taking shortcuts? Because we know that the data says that it's actually a 2-dose vaccine, that the second dose should be given after 21 days, and that only they tried to give 1 dose in the phase 1 and phase 2 sort of trials, but these are not peer related trials. So what's going on there? Is this all political? Is this all something that's just to get the votes up or to win the race?
It's a burning question, I think, on a lot of people's minds. And I believe the clinical trial data was from about 36 vaccines, so it's a really small number. And to my knowledge, this is an adenovirus, which expresses a spike protein. There are a number of vaccine candidates that are taking a similar format. And I believe they're coming with adenovirus serotype 5, which is the same one that was actually used in the Merck Step 5 HIV vaccine. So, you know, there's certainly a priming event, and then they'll need to test out a boosting. And I think the boost will be with another vaccine format. So they're looking at giving one attenuated virus for the priming, so for the first shot, and another vaccine for the second shot. You know, really, to be honest, the numbers are way too small to say whether you're inducing good immunogenicity, so whether the vaccine is actually inducing a robust response. And also, you're going to have no data around toxicity or you know, disease enhancement. So it's really too early, and I think this is certainly a political move on the part of Putin. You know, we're going to wait to see what happens, but it's really a crazy scenario.
Let's see who gets elected first, Trump or Putin.
Yeah, absolutely. And China, you know, of course, they're left out for various reasons from Operation Warp Speed. But out of the 6 candidates, I believe 3 of them are Chinese, and they're moving forward. And some of these Chinese candidates are also being tested, you know, for instance, in Brazil and a number of other countries. So, those are certainly moving forward as well. And the data is looking quite compelling as well. But really the proof is in the pudding. We really need to see tens of thousands of individuals being vaccinated in hotspots like Brazil, like in the US, where you can really see how and whether it has an impact on the infection numbers. So I think I have one more slide. Anton already alluded to this, and this is really the need, during a global pandemic for there to be equitable access to vaccines. And there's actually an organization which is called COVAX, and this is made up of a number of different organizations like the funding agency CEPI, Gavi, and also the World Health Organization. And COVAX is essentially the largest and most diverse COVID-19 vaccine portfolio in the world. It comprises 172 countries and a number of candidate vaccines which are engaged in this particular initiative. This actually is made up of 80 potentially self-financing countries. South Africa is actually a part of these 80, and 92 low and middle income economies eligible to be supported. Up to now, 9 candidate vaccines are part of this initiative, and another 9 candidates are actually under evaluation. And the goal of COVAX is actually bringing the pandemic under control via equitable access to COVID-19 vaccines, broad-scale commitment, and also investment from countries. So one hopes that such an initiative will really have an impact in taking an efficacious vaccine to the world, and ideally to the developing world. So, it's not just the wealthy countries that are benefiting from COVID-19 vaccines.
Yeah, I must say, Brian, that was an absolutely excellent talk. I mean, it really makes sense to us what's actually going on, how the vaccines work, why they work, the different types of vaccines that are available, what's in R&D. And I just want to ask you some burning questions that are on people's minds. You spoke about the neutralizing antibodies. Can you explain what neutralizing antibodies are? What do they mean? And what's the difference between neutralizing antibodies and T cells? We know that the antibodies wane in COVID, but T cells are supposedly supposed to actually remain for a number of years. What's your thoughts on this?
So, Anton, that's a really important question in the context of vaccine-induced protection. And as I mentioned, a lot of vaccines are actually targeting the generation of neutralizing antibody responses. And neutralizing antibodies are produced by B cells. And these are antibodies or proteins that have the capacity to bind to a virus, in the case of SARS-CoV-2, and prevent infection of the host cells. So you can imagine that the neutralizing antibody will actually bind to the surface. In this case, it could be the spike protein. And now the spike protein is not available to bind to the ACE2 receptor on the host cell. So it actually prevents an infection event. And that's what one really hopes to see. At the same time, I think there's really a need for a T cell response. So T cells are really coordinating cells of the immune system. The helper cells are really involved in also driving a really good B cell. So evolving the response so that you get better neutralizing antibodies and also different types of antibodies that can have different functional outcomes. Helper cells are also really involved and important for persistence of immunity. So in people who don't have helper cells, you actually see that the immune response actually dwindles a lot faster. And then I also mentioned killer cells or cytotoxic T lymphocytes. These are really important to recognize virally infected cells and to actually eliminate them from the system. So really there's going to be a need for both of these arms of the immune response.
Brian, can I just ask you, effectively what you're saying is that the vaccine either prevents infection or it fights infection, those in simple lay person's terms. How does a company or an institution or an institute decide which path they're going along?
Yeah. Well, I think, first of all, I would say, you can really have protective or what we call prophylactic vaccines, where you vaccinate a naive population and you hope that you can actually prevent them from getting the disease, and on the other hand, you can actually have therapeutic vaccines. And these are vaccines where you actually vaccinate a diseased population and you hope that you can ameliorate the disease and ideally actually cure. And this is the direction HIV is going in. We still don't have one, but the concept of cure is now on everyone's minds. Can we actually have a vaccine that can actually not only reduce the disease and lead to an undetectable thing, but actually eliminate it from the system? And I think it really depends on the nature of the infection. Is it a chronic infection that's around for a long time, or is it something acute, like in the case of a flu, where the virus is usually cleared in about 10 to 14 days. With COVID, there's still a lot of unknown answers. How long it persists for. It seems like it's in an acute situation, but we are seeing scenarios where people have got symptoms that are lasting for a long time. And I know also a lot of people are really concerned about, aspects like reinfection. I think it's still really early, but, at the end of the day, we really need an effective vaccine. At the same time, therapeutics or antibodies are under development, and hopefully some of these are also going to play a role in reducing the severity at the moment until we actually get a vaccine. So, there are a number of sort of strategies which are ongoing at the moment, and hopefully we're going to achieve the sort of 12 to 18 months. I think the challenges mentioned will also be scaling up the vaccine candidates, to really cover a world population of 7.8 billion is not a trivial thing to do.
So let me ask you my final question. What about convalescent plasma? Okay, convalescent plasma, which has now been sort of authorized as emergency treatment by the FDA. And what does it do to your antibodies? What does it do to your T cells? And does it affect you getting a vaccine? Can it affect your further protection from that point of view? It's a treatment, but does it affect further your actual immunology of your body?
Yep. So we're actually doing a study with an academic institution in the US where they are looking at convalescent individuals, so recovered patients, approximately 1 month since they've been symptom-free. They're taking blood from these individuals, measuring the binding titers against SARS-CoV-2, and then these plasmas are then being used to infuse infected patients in the hope that you can transfer what we call passive immunity. So we're hoping that within those plasmas from the recovered patients, they're neutralizing antibodies that have the potential to bind to the virus and actually help clear it. So that's the basic concept of plasma therapy, and it really goes back decades. It's a strategy that's really used to be expeditious. Because it's passive immunity, one will envisage that it will stay around in the system for a few weeks or maybe a number of months, and then will be eliminated from the system. And it's not inducing an immune response itself because of the passive nature. So we wouldn't expect it to have an impact on vaccine response. Anton, another important point I wanted to mention was a recent study actually looking at SARS infection. And going back about 17 years, they found that antibody responses were detectable for about 2 or 3 years, and were then not actually detectable after about 17 years. But they were able to detect T cell responses in some of these individuals. It was really small numbers. I think it was somewhere around 3 individuals, or 3 to 5 individuals. But was really interesting and important to note is that you may have an antibody response dwindling, and there may still be a T cell response around. And there's a lot of concern around, the antibody responses actually dwindling really quickly after infection. And I think, there's still a lot of data being gathered around this. But we also need to look at the cellular response at the same time and understand how long-lived CD4 and CD8 T cells are, because that's also a really important arm of the protective immune response. Another important thing to note is also potential cross-protection. We don't know at this stage, but there are about 4 different common cold viruses, which are actually coronavirus in origin. And some of these lead to cross-reactive T cells in individuals, and these have been detected and published in a number of studies. And we don't know how this is going to impact infection or even vaccine outcomes, but it's something really important to note as well.
Anton, your thoughts and your feedback. Are you more hopeful around where we are in terms of vaccine?
I mean, you've alluded to a lot of new facts here and a lot of interesting facts, and I definitely am more hopeful, especially even with this last point you made with regards to the cross-reaction with the coronaviruses themselves. You know, there's a lot of people that are talking about it and saying that if you have had a coronavirus in the last 3 or 4 years, potentially that if you do get this cross-reaction with COVID-19, you will get a less symptomatic type of illness, but that remains to be seen. With regards to the COVID-19 vaccine, we need it now. There's no doubt about it. We need to squash this virus, and we are very hopeful that within the next few months we will get it, and hopefully it's efficacious, and hopefully it's equitable, and hopefully we can get it across to as many people as possible.
Brian, are you, I mean, you're involved in this and you're involved in a company, so you obviously clearly do believe that within our 12 to 18 months, as you've mentioned, that we should see a vaccine.
I am hopeful. As I mentioned, you know, the proof is in the pudding, but never before in the history have we seen so many candidates under development. The fact that in the space of about 7 months, we have 6 candidates moving into phase 3 is unparalleled. And, you know, one will hope that a few candidates will kind of move forward, show efficacy, won't be, you know, cause adverse events. And I think importantly, as mentioned, we'll need a number of candidates to really cover a world population. So, yeah. Like everyone, I certainly hope that we can achieve that in the sort of 18-month time frame.
Absolutely. And thank you both. Thank you, Brian Abel. Thank you, Anton Meyberg, for what I think was the most incredible discussion. Hugely informative, very, very interesting indeed. And I have no doubt that we'll be regrouping for a reunion sometime in the next few weeks to see how these things are evolving and developing. I'm Howard Feldman. This has been the Synthesis of Podcast. Please make sure to subscribe to our YouTube channel in order to make sure that you get the alerts when new podcasts are posted. Be safe. Have a great one.